We have talked about beta-lactam antibiotics, which inhibit bacterial cell wall synthesis. Changed the world because now people didn't die of wounds and injuries quite as much as they used to. Dramatically changed the course of lives of soldiers in World War II. We've talked about NSAIDs, which are pain relief, and thankfully non-addictive, inhibiting the COX-2 and COX-1 enzymes that make prostaglandins and result in pain and inflammation. What we'll start on today by request of some of you, I did, yep, is the hormonal contraceptives. So there are a lot of ways to do this, but many, and we'll talk about combination oral contraceptive pills versus progesterone-only pills. But two main components for contraceptives are a synthetic estrogen derivative and then a synthetic progesterone. So if you didn't know, this is the structure of estrogen. You've got a benzene ring, and it has sort of the core structure of a lot of steroids. It's made as a derivative of cholesterol. It's got three 6-membered rings fused together, tons of stereocenters. A lot of steroids share the same core structure. And then for estrogen, or this is called estradiol, there are other forms of estrogen that the body uses, but this is estradiol. This is the one that is natural. And then a common estrogen derivative that's synthetic basically has almost exactly the same structure, only it adds an alkyne group right here, and that helps with its bioavailability and how it is metabolized and how long it lasts. So this is ethinyl estradiol, and as you might expect, based on the name, the target for the synthetic estrogen is the estrogen receptor. And this particular molecule is called an agonist of the estrogen receptor. There's, when dealing with molecules that interact with receptors, there's some interesting language. You've got agonist, which means it activates the receptor. And then you've got antagonist, which means it prevents the receptor from doing its thing. And there's multiple ways that can happen. But for an agonist, it binds the estrogen receptor in the place of estrogen, and it elicits the same effects as estrogen. Once you bind the estrogen receptor, the basic intended action, originally estrogen was included in oral contraceptives to prevent what's called breakthrough bleeding, which is spotting and bleeding not at the expected time during the cycle. So it was originally included to prevent breakthrough bleeding, but it also inhibits ovulation by reducing the secretion of, I'm just going to abbreviate FSH, which is follicle stimulating hormone. And as we talk about that stuff, we're already well outside my area of expertise. Don't really have a lot off the top of my head in terms of the biochemical mechanisms of reproductive biology. So I know that follicle stimulating hormone is involved in stimulating follicles resulting in ovulation, but that's about all I know. So you'll have to rely on your other bio classes to give yourself some more information there. Then the other component of hormonal contraceptives are derivatives of progesterone. And it's really interesting, if you look at the structure of progesterone, you'll see that it's similar to estrogen, but it has key differences. Instead of this benzene ring here, progesterone's got a 6-membered ring with a ketone on it. Other than that, a lot of the core structure is similar, especially the fused 6-membered rings. And then another 5-membered ring here, a methyl group there, and this time instead of an alcohol, a ketone. So that's progesterone, that's natural progesterone. And then here are some examples of some synthetic, they call them progestins, synthetic derivatives of progesterone. You can basically take the same progesterone structure and then mess around with it just a little bit, add a methyl group here, an OH group here, and another alkyne. And this is called levonorgestrel, and that's a very, very commonly prescribed progestin. And then an alternative is called, let's see, norethindrone. And that'll be exactly the same, except it lacks the methyl group that levonorgestrel had. And one of the things that should emerge as we look at structures, and I'm not really concerned about you memorizing detailed structural features. For example, do I care that levonorgestrel has an ethyl group here, whereas norethindrone only has a methyl group there? No. But if you looked at a steroid like this, or like this, if you could recognize it as a component of a hormonal contraceptive, if you could recognize these as being related to estrogen and these as being related to progesterone, that would be enough. And in fact, the big difference between estrogen and progesterone is the benzene ring in the alcohol versus this 6-membered ring with the ketone. You got that? That's basically all you need to know. And of course, I'm not going to be saying on this exam, draw progesterone, because you won't need to draw anything. It will be a multiple choice quiz. So which of the following is a progestin, or which of the following is estrogen? If you can focus in on that ring on the left-hand side that's either a benzene ring or has a ketone, that should be good enough. The target for these progesterone derivatives is, of course, these are all agonists of the progesterone receptor. And if you want to learn more about membrane receptor biology in cells and tissues and organs, that'll be another class with people that are more qualified than me to talk about. But the action of activating the progesterone receptor is basically to inhibit ovulation. Two main mechanisms: inhibit ovulation. The way this works is it's a decrease in gonadotropin releasing hormone, which decreases the presence of FSH and luteinizing hormone, and that prevents ovulation. And another mechanism is thickening of cervical mucus, which prevents sperm from being able to find the egg because they can't move through the mucus quite as quickly. So those are the structures, targets, and main actions of both of those. It's interesting, there's a lot of different varieties in terms of how you can take this. The combined oral contraceptive pill has both the estrogen and the progesterone in it. And the medication, the regime for taking it is about 21 days of the pill that you take and then 7 days that you don't. In many formulations, there's a placebo pill for the remaining 7 days that you take, just so that you stay so that you're in the habit of taking a pill every single day. There's also options for a progesterone-only pill. And you'd have to talk to a physician to understand why you'd want to take progesterone only versus the combination pill. Progesterone only pill you take daily within the same 3-hour window. There's a lower dose mini pill that you can take that inhibits pregnancy mostly by thickening cervical mucus. Higher levels of progesterone also inhibit ovulation. Perfect use, meaning you take the pill every single day as indicated, has a success rate, we'll call it a failure rate, though that seems negative because babies are wonderful, but a failure rate of around 0.05 to 2% pregnancy occurrence per year if you've got perfect use. And the 2% is probably a little bit high. They advertise more than 99% effective. Typical use, meaning what most adult humans, perhaps in the United States, are able to pull together, it can be as effective as 90%. And again, this is probably if you forget. So a failure rate of about 9% pregnancies per year. So can be effective if properly used. Side effects, as I said, include bleeding, nausea. Nausea is a side effect for just about every medication. It's unclear exactly what's going on there. Headaches, abdominal cramping, breast tenderness, and some others that you can read about. Anything you can ask me. I've basically told you what I was able to figure out. Any questions you have about that? Go ahead.
This probably has zero relevance, but how do names change so much from for example, the methyl group from lemon progesterol to
right.
How does the name change that much?
That's an interesting question. How are drugs named? I mean, you learned some nomenclature rules way back 100 years ago when we started this class, right? And the IUPAC name for this would have been horrible. And so some people to name molecules use a traditional approach that's happened basically as we've discovered new molecules. Like, why is cholesterol named cholesterol? No idea. Why is estrogen and progesterone have those names? Well, at least progest sounds like favoring for pregnancy or gestation. In terms of why these have these particular names, levonorgestrel and norethindrone, so when a drug is developed by a company, there's two different names they give it. They give it the name of the molecule that they patent it with, and then there's a trade name that they market it with, and those two names are different. So Tylenol is the trade name, acetaminophen is the name of the molecule. And we could go back and look at the NSAIDs, and some of those names make sense, like Ibu was the isobutyl group. But when it comes to larger molecules, the actual molecule name, it sometimes means something, and then sometimes they do focus groups to see whether the name sounds scary or not. So levo probably refers to dextro versus levo, left-handed versus right-handed. Molecules that have levo in it are chiral and probably rotate light in the right-handed direction or something. Gest probably is for progesterone. Nor, I'm not sure. The nor usually means that it's missing something. And other than that, I got nothing. Norethindrone, there's the nor. I bet the eth is related to the ethyl group, so there used to be an ethyl group there, now there's not. And then the one probably means ketone. But yeah, it's really, really weird. And literally the people that make the chemicals usually name it like the abbreviation of the chemical company and then some number code after it. And then once they've done the tests, then the business people get involved and they're like, what should we call this? What sounds good? Does norethindrone make you feel scared or sciencey? I'm in the hands of good scientists or not? So yeah. Okay, good. Other questions? Okay. Good. All right, so now we're going to talk about statins. Statins are, if you look at the top 20 prescribed drugs worldwide in 2022, many of them are statins. The top one is a statin. And statin, I'm not sure where that particular name came from. It's probably related to stasis or level of something. These are for, and this is what doctors and other medical professionals like. Why do you take a drug? That's called an indication. You take a statin because of high cholesterol, particularly LDL. We didn't have a chance to talk this semester about HDL and LDL. You can do some reading about that. LDL is the bad kind of cholesterol. LDL is a low-density lipoprotein particle. It's like a micelle that transports lipids in the blood, including cholesterol. And LDL deposits cholesterol in tissues, whereas HDL picks up cholesterol from tissues, takes it to the liver, and it can be metabolized and gotten rid of there. So LDL is the bad kind, and statin drugs lower LDL. And this is, again, the most prescribed drug in the United States are statins. And it prevents, in addition to high cholesterol, it's thought to prevent atherosclerosis, which is depositing of cholesterol and lipids in arteries and hardening of the arteries, preventing atherosclerosis and cardiovascular disease. So you guys are all young, but at some point as you get in your 30s and 40s, your physician will do a blood test and say, hey, I don't like how high that cholesterol is. And your physician will say, you can try to control this through diet and exercise, and you maybe you'll do that, and that's a good thing to do. Then you'll come back in and he'll say, great, you're doing great. Or he might say, he or she might say, you know what, it's still high, we really ought to get you on a statin. And the idea of the statin is it lowers your cholesterol and then prevents cardiovascular disease. So a scientist once came to the University of Wisconsin, where I was getting my PhD, and gave a talk and said that chemists don't typically know how to communicate what they're doing in ways that people understand. Like, I'm going to draw for you this statin drug, like several of these statin drugs, and you'll be like, huh, okay, that's nice. But actually, he suggested that we say statins change the way we die, because we're no longer dying as frequently in our 50s, 60s, and 70s from heart attack and stroke because of the statin drugs. So let me show you what some of these look like, then we'll talk about how they work. The first statin that was discovered was called lovastatin. It's actually a natural product. It's made from a soil mold fungus. And it's also naturally found in red yeast rice. It's got 2 6-membered rings, double bonds there, hydrogen there, methyl group there, a weird kind of side chain here that has a stereocenter, and I'm already running out of space. Then another 6-membered ring here in which one of the atoms is oxygen. A methyl group here, and then there it is, an oxygen and a side chain. So pretty complicated. This is a natural product, so they could isolate it from the soil fungus. This was approved by the FDA for use in managing cholesterol in 1987. So that's within my lifetime. I was baptized that year. So before, we didn't really have a way of managing cholesterol. And then starting in the 80s, we did. A related molecule very similar to lovastatin is called simvastatin. And this is just, you'll look at it and be like, how on earth did they decide, hey, let's take this thing from mold and let's just add a methyl group on it? And it would be an interesting question as to whether simvastatin was better or worse than lovastatin. This was developed by the drug company called Merck. It was approved in 1992 by the FDA. So you can see the late 80s and 90s were important. This drug had the trade name Zocor. So simvastatin, again, no attempt there. Simvastatin, where did that name come from? I got no idea other than sim. Maybe it's similar to lovastatin. You know? Zocor, and that's the trade name, and that's purely the marketers coming up with something they think sounds cool enough that Americans will think it's powerful and good to take, but not weird enough or complicated enough that they can't say it. Because if you're supposed to ask your doctor if Zocor is right for you and you can't even say Zocor, then you probably got problems. Some of this has gotten way out of hand. Like I am in my 40s, and when we watch Hulu at night, we get all kinds of advertisements. Hulu knows our demographic, and we get all kinds of advertisements for autoimmune disease type treatments. And I don't know why I would ever ask my doctor if Sky-Rizzy was right for me. That's just bizarre. Like, who came up with that name, right? Like, maybe it was a teenager. Like, this will give you lots of Riz, man. Yeah. Okay, so interestingly, I'll show you one more statin drug. It's called a statin because it does the same thing as these guys do up top, but its structure is dramatically different. And all of these drugs bind the same active site in the same protein, which I will tell you about in a little bit. Atorvastatin, which I'm actually on this one, so this is one of the first drugs that I'm currently taking that I'll tell you about today, has a 5-membered ring core with a nitrogen. That's called a pyrrole. And then it's got a branching arm over here with several OH group stereocenters, and then a carboxylic acid. Oops, there we go. But we're not done yet. There's more. And there is a benzene ring here that has a fluorine, and there is another benzene ring here, and then yes, an amide, and another benzene ring here. So you might ask, how on earth was that invented? If they knew this molecule did the right stuff, who in their right mind was like, let's make this one? And this is what medicinal chemists do. People that get trained in synthetic organic chemistry go to drug companies and they learn how to, they get what's called a lead compound that has some activity, and then they start modifying it and trying to get better and better effects. Along the way, you can see that somebody was like, well, what if we put a fluorine there? What would that do? And that apparently made it a better drug. So atorvastatin, the story is interesting. It was made by a company called Warner Lambert in 1986. It was approved by the FDA in 1996, and it was co-marketed by the company Warner Lambert, which no longer exists, and Pfizer, because Pfizer bought them out for $90 million? And the drug was Lipitor. And Lipitor is the #1 prescribed medication in 2022. Over the course of its patent lifetime at Pfizer, it made Pfizer $125 billion. Now, thanks to folks like Elon Musk and Apple and all these companies, that no longer sounds like a lot of money, but that is a huge amount of money for a drug company. $125 billion over 14.5 years, and in its heyday, $13 billion a year. It accounted for about a single drug accounted for about 25% of Pfizer's revenue. So this is where the term, if you've heard blockbuster drug, comes from, because it made Pfizer a ton of money. Yeah.
What about things like vaccines? Just thinking of like Pfizer shot of like COVID shot, and so would, for example, would a vaccine look something look like a molecule like this?
Yeah, so how do vaccines work? That is a good question. In general, vaccines are not small molecules. Vaccines are often much larger molecules, and we could say a ton about this, but I don't actually know a ton about this. Multiple ways you can do vaccination, but the idea is that the pathogen, the virus or the bacterium, has some kind of group of molecules on its surface or inside that don't belong in you. And the purpose of a vaccine is to teach your immune system to recognize that and then neutralize it later. So my colleague Paul Savage could tell you a lot more about this than I could. The COVID vaccine was an mRNA-based vaccine. In other words, it didn't have any virus in it, but it had instructions for making viral proteins. So the COVID vaccine, you had to formulate the mRNA inside, I think that's called a liposome, which is something of a membrane that helps the mRNA helps the vaccine to fuse with your cells and dump the mRNA inside your cells. The cells that get that mRNA then start making viral proteins, not any that are going to actually make virus, but I think it was the spike protein on the COVID particle, and that would get translocated to your cell surface, and suddenly your antibodies are like, that doesn't belong there, and that triggers a process where your body does basically what's called affinity maturation, or it does its own directed evolution experiment where it changes the sequence of the antibody until it finds one that binds really tight, and then your body can now make that antibody, and it can now clear virus, because viruses make those proteins too. A lot of different strategies for vaccines, but that's the basic idea. Can you elicit an immune response so that your body will make antibodies to the bad stuff? The side effects of most vaccines, which include fever and chills, are because your immune system's being activated. Not that you're getting sick, but that your immune system's getting activated. Okay, let's see. So what does atorvastatin do? What do all of these statins do? Their target is an enzyme called HMG-CoA reductase. And I'll show you why that works, but the effect is to lower LDL and thereby prevent atherosclerosis and cardiovascular disease. Side effects can include muscle pain, though it's unclear based on what I was able to read how prevalent that is. In fact, there was at least one study that suggested that that the muscle pain sometimes associated with statin drugs was what they called a nocebo effect. You've heard of the placebo effect, which is where you take something that has no drug in it, and yet there's an effect because you expect it to be. The nocebo effect is where you anticipate that the drug is going to do something negative to you, and then lo and behold, it does. So they're advising physicians not to warn too much about muscle pain because they think it's maybe not that big of a deal. In any case, let me show you a couple of metabolic pathways. You don't have to memorize these. I just want you to have an idea of what's going on. This is the mevalonate pathway, and this is a precursor to cholesterol synthesis. So this is just lifted from Wikipedia. It starts with acetyl-CoA. You do an aldol reaction or a Claisen reaction to get two acetyl-CoAs together, and then a third aldol reaction to get this molecule called HMG-CoA. And then it gets reduced by this enzyme HMG-CoA reductase. That's the enzyme that gets inhibited by these statin drugs. Now, this is not even close to the cholesterol end product. This is what's really interesting about this. You inhibit something that's very, very, very early on. By the time the pathway's over, you get to these two molecules here, called isopentenyl pyrophosphate and dimethyl allopyrophosphate. And boy, do I wish I could tell you more about those. We spend like 4 or 5 days on this in when I teach Chem 352. It's awesome. There's all kinds of really cool chemistry. But let's just say that these molecules are very far away from cholesterol, right? They each have 5 carbons. Cholesterol has like 30. So how does that work? Let me just sketch this out briefly for you. The molecules IPP, isopentenyl pyrophosphate, and dimethyl allopyrophosphate get turned into a much larger molecule called farnesyl pyrophosphate, which has 15 carbons. Then that gets turned into a molecule called squalene, which has 30 carbons. Squalene eventually gets cyclized to something called lanosterol, and then there are like 19 more steps, and then you finally get to cholesterol. So, massively long process, inhibited really early on, and it has the effect of lowering cholesterol. Apparently without messing with a lot of the other hormones that are also derived from cholesterol, which is kind of interesting. Okay, anything you want to know about statins that we haven't talked about? Oh, I wanted to show you, there's a crystal structure of atorvastatin bound in the active site of the enzyme. So you can see those benzene rings are doing things. I haven't shown you any of the protein side chains because that would make things more complicated. But if I did, you would see that these groups are making contacts with other side chains, non-covalent interactions, hydrogen bonding, dispersion forces, and so on with groups in the enzyme active site. So that molecule binds in the place of the desired substrate, HMG-CoA, prevents it from getting reduced, and then that reduces the synthesis of cholesterol. Yeah, anything else? Yeah.
I was just wondering how, when going over these, how can we know which group goes in? Like, for instance, for the beta actins, we know which ones they are because of the box.
Right.
So how can we look at,
How do you recognize statins
yeah,
based on structure? Okay. These two are very similar. They almost look like a steroid that's been broken apart. You have 2 fused 6-membered rings together and then another one hanging out up there. Atorvastatin, however, is unique, and I'm not sure how to remember that one. Yeah. Forgot?
No, I just, what I was going to say was incorrect.
Okay. Yeah.
When people are synthesizing like new drugs, how do they know that it will have the intended effect without testing it on a human?
Great question. When you're doing drug discovery, how do you know that it will have the intended effect before doing it on a human? Well, this is where you need what is called an assay, which is a way of measuring whether or not, or assessing whether or not the molecule has a desired activity. In this case, you could have envisioned 2 different things. First, a binding assay. There are analytical ways to determine whether a molecule binds another molecule. So you can measure the strength of how tightly it binds. The other thing you can do is a functional assay where you have this enzyme that has an effect and you measure that effect and then you add your inhibitor and you see if that effect goes away. That's called an in vitro assay, which means not in a living organism, in a test tube. So that's a biochemical assay. Usually after you've demonstrated that something has effects in a biochemical assay, then you start doing other tests or other more complicated systems, including animals. So you might have a mouse model for hypercholesterolemia or whatever, having too high cholesterol. You administer the drug to the animal and you determine whether or not it has an intended effect. At some point, you start being able to do clinical trials in humans, and there are various phases of those clinical trials. I forget, and somebody who's smarter than me would know, but phase 1 and phase 2 are related to safety, safety and toxicity. You have to demonstrate before you can even put it in people who are sick that it's basically, you have to determine what the side effects are and how the drug is distributed and metabolized and so on. And then it's later on that you have to do phase 3 clinical trials, usually double-blind controlled studies, where you demonstrate that your molecule actually changes patient outcomes relative to what other approaches are currently available. So yeah, it's a really long process, extremely expensive, and most drug development efforts fail. And so this is one of the reasons we could talk about why drugs are so expensive, but one of the reasons they're so expensive is the successful ones have to pay for all of the failed efforts. All right, good, yeah.
So how do you get the drug to like dissolve and metabolize in the proper location? How do you target? Like, is it supposed to spread throughout the whole body? Is it supposed
Right.
to target?
These are great questions. The issues that you've brought up are basically bioavailability. That means, well, actually, we could, even before we put bioavailability, we could do administration route. That is, how are you going to get it in the people? Second, bioavailability and distribution. That's called, sometimes people call that pharmacokinetics. That's like, how readily does it dissolve when it's in your stomach? Or, and then from there, how does it get distributed? Does it cross the blood-brain barrier? Does it accumulate in any particular organs? And then there's also metabolism and elimination. So, does your body do any chemistry on this molecule? A lot of metabolism of drug molecules happens in the liver. A lot of it is related to oxidation, which helps the molecules being eliminated. And then, yeah, how do you get rid of it? Does it get excreted through urine, or is it eliminated in some other way? All of those are really important issues. With metabolism, you're worried about, are any of the metabolites toxic? So a lot of key questions, and you have to do those experiments too. For most of the molecules we've been talking about, you take them as a pill, so that's oral administration. It's got usually the pill you take is not the pure molecule. It's formulated with other things to help the molecule be in a form that can actually exist as a tablet and not fall apart into a powder. Easy to swallow, but once it gets in your stomach, will dissolve. Of course, maybe you want that process to be a little slower so that it's not as soon as you put it in your mouth that it starts to dissolve, or maybe that's the desired outcome. A lot of different things that people worry about. There are sophisticated drugs that are targeted towards specific tissues. These usually involve conjugating a drug to a molecule that binds some other particular thing in a certain tissue, and that can be highly effective. It's kind of hard to do. So I just basically told you there are a lot of issues to follow that I don't know about.
Yeah, that's cool. I wouldn't understand a little bit if you didn't know.
These issues of metabolism and elimination affect the dose rate, like how often you have to take a pill. The dosage rate for statins is basically 1 pill daily. There are some drugs you don't have to take that frequently. Okay, what else? So let me just, I'll tell you the drugs that I'm prepared to talk about, and then you can pick the ones. I think we've done only hormonal contraceptives and statins. I'm prepared to talk about ACE inhibitors, which are for blood pressure, beta blockers, which are sometimes for blood pressure, but also to prevent secondary heart attack, to prevent heart arrhythmia. We can talk about SSRIs, and we can talk about opioids. So we might not get to all of them. So which one do you want to do first? Opioids? All right, you chose it. Be prepared to draw some cool things. These are wacky, complicated molecules. So let's take a break and we'll pick up with opioids after the break.
All right, let's do it.